Hey again, Beaks and Beaklings, it's Mr. Boylan, and today I'd like to draw out meiosis with you. Okay, hopefully you've already studied mitosis or watched my video on mitosis and feel comfortable with that, because meiosis is a lot easier to understand once you understand mitosis. Okay, although they're very different processes. And again, when I'm drawing today, I'm probably going to use red, yellow, blue, and then something to write with again. So I wanted to start by talking about what meiosis is, okay, and compare it to mitosis. So last time we did mitosis, and this was asexual reproduction, right? This is when we had one diploid cell. So for example, a 2N cell, and in humans it was 2N equals 46, and we created two genetically identical daughter cells, and these are both diploid. One of the hardest things for students to understand is, wait, how did we go from 46 chromosomes to two cells with 46 chromosomes? Because these 46 chromosomes look like this, and remember, when you split this into two daughter chromosomes, you have 46 of these. Okay, so that was mitosis. That's asexual reproduction there. What we're going to talk about today is meiosis. Okay, and what this is you're actually creating dissimilar cells. You don't want them to be identical. These two cells are identical. If I fall and scrape my knee, I better have the exact same types of cells genetically fill in the gaps there. But in meiosis, we're actually going to do two sets of division. Now, this is not part of sexual reproduction here. This is actually making what we call gametes or sex cells so that you can do sexual reproduction with these cells. Okay, so we're still just going to start with a somatic body cell. Okay, notice I use the word somatic there, somatic body cell. And this is a 2N equals 46. And what happens in meiosis is we have two meiotic divisions. In meiosis 1, we went from a 2N equals 46 cell, but now we have actually haploids. This is very weird. N equals 23 and N equals 23. These, you're going to find out, actually are useless. Okay, or they're not useless, but they're not finished yet. So then we go through meiosis 2, and we're going to now create 4 dissimilar haploid gametes, and all of these cells are going to be N equals 23, okay, and they are dissimilar, which is great because we need a lot of variation as sexually reproducing organisms. That's why we're still here today, probably. We have a lot of variation in our population, and we'll see some of the reasons why today. Okay. Now, another thing to consider here is that guys and girls do this process a bit differently. Okay, so if we're doing meiosis, let's talk about a guy. Okay, pretty much what we just drew. A guy starts off with a somatic cell. Okay, as a human being, we are diploid. We had a mom and dad, right, and each gave us 23 chromosomes, so we're a diploid 2N equals 46 cell. In meiosis 1, we split to N equals 23, two of those cells. And you might be wondering how that can happen. Do you remember how you have double-stranded chromosomes, single-stranded chromosomes, or daughter chromosomes? In this case, we're going to be separating pairs, so bear with me. It gets understandable eventually. And then both of these are going to go ahead and differentiate into 4 dissimilar N equals 23 haploid gametes. Okay, and this is because you're going from a double-stranded chromosome and ripping it apart. So that's how the number stays the same there. And obviously when gametogenesis happens, when you create these cells, and this would be in a guy's testes, right? He's eventually going to make these into sperm. Okay. Girls are a little bit different the way that they do it. So what she does is she starts off with 2N equals 46 in her ovaries. But before she was even born, okay, while the girl was in her mom's womb still, she actually started this process. But do you notice what she did here? In meiosis 1, she still has an N equals 23 cell here, but she stole most of the cytoplasm this side. And then in meiosis 2, she does it again. So technically, n equals 23. She creates these three kind of waste cells, okay? And we call these polar bodies. So a girl will make one, eventually when she hits puberty and goes through ovulation, a girl will make one good gamete, okay, or a sex cell. You start with somatic and you end up with this one egg here, okay? So these were sperm and gametogenesis for a girl. She's going to make an oocyte or an egg. These three are just waste polar bodies. They're not used for sexual reproduction. So why did she steal the cytoplasm? Probably because the egg is so important, right? There's an old, now debunked thought of ex ovo omne vivum in Latin, from the egg everything. The egg is really, really important. It's so important. As a matter of fact, when fertilization happens, we're going to have an egg and we're going to have the sperm coming in here, okay? But that egg, when the zygote forms, the zygote's basically just a fertilized egg. There's a lot of nutrients and things in here for that growing embryo, okay, or eventual growing embryo. One other thing I wanted to point out before we go ahead and draw the phases of meiosis is that, sure, the somatic cell that we started with had to go through G1, S, and G2 and M, things like that. But once you decide to trigger meiosis or a meiotic event, you are no longer part of the sex or the cell cycle. When you're making a sex cell or a gamete, you literally go through gametogenesis to create the gamete, sperm or eggs, and those cells stay there until fertilization. Once fertilization occurs and you form a zygote, which in this case, n equals 23, n equals 23, two haploids would come together and form a zygote, which would be 2n equals 46. And this zygote will now go through mitosis for the rest of the organism's life until you can start doing meiosis and making sex cells all over again. Okay? So let's go ahead and draw the phases of meiosis. Okay, so if you watch my other video, some textbooks use a pro-metaphase during mitosis. Meiosis, we just get rid of that. So in meiosis, we have meiosis 1 and meiosis 2, just PMAT. Prophase, metaphase, anaphase, and telophase. Okay? So here we go. Prophase 1 is probably the most important phase of meiosis. And if you learned at all about gene linkage and things like that, super, super important what happens here. There's a lot of why we're so different from each other that occurs right here. And it's so crazy that you can't even calculate it, to be honest, because it's very random what happens. So here's a cell, and let's just say we're doing male meiosis right here. And you remember in prophase, the hallmark is that, oh, okay, we coil up chromosomes. So I am not going to draw 46 chromosomes just like last time, but I will draw, let's say, 4 chromosomes. All right, just like last time. So we're going to say this is a 2n equals 4 cell. If this was a human, it would be 46. We're just going to draw 4. And one of the first things that happens is the chromosomes start to coil up. However, little different here. As these chromosomes coil, so the chromatin fibers, right, are starting to coil up, something interesting happens. There's number 1 from my mom, and let's say here's number 2 from my mom. Okay. What happens is synapsis. Number 1 from my dad can line up right next to my mom's number 1, and number 2 from my dad also lines up right next to my mom's number 2. What's happening right here is called crossing over. This is so important, okay? And we're going to come back to this at the end and make sure we understand it. But there is, of course, nuclear envelope will eventually break down, right? There's all those other organelles in here. We're not going to worry about them. We do have centrosomes with centrioles in them, right? Pairs of centrioles. So here's a little centriole pair in there and there. Aster, here's an early mitotic spindle or meiotic spindle, right? So there we go. So prophase 1, what happens is we have 1, 2, 3, 4 sister chromatids lined up. And what we do is we call this a tetrad. So a tetrad forms, okay? And these form during, okay, synapsis. Now what synapsis is the pairing of homologs. And if you don't, excuse me, remember what a homolog is, or a homologous pair, or a homologous chromosome set, you can go back and watch my other video. But basically what it means is that the genes on both of these chromosomes right here are the same, right? The genes are the same. Are the alleles the same? Oh, well, maybe not. Maybe my dad gave me free earlobes, but my mom gave me attached earlobes. The gene is the same, right? We all have the same genes, but the alleles could be different because the sequence of DNA can be different, okay? So what's going on right here, okay, or right here, okay, this is called the chiasma, okay, so chiasma, or of the cross. So this is where crossing over, okay, occurs. So that chiasma crossing over occurs. And this usually happens like 2 or 3 times per chromosome pair. There's some crossing over that goes on in here regularly. And if you think about it, you're going to exchange corresponding segments. No big deal. For example, oh no, I'm changing the earlobe attachment gene from my maternal chromosome with my paternal chromosome. Okay, it's still the earlobe attachment gene. So crossing over, really what it's doing is it's taking the chromosomes that you have and shuffling them up. Okay, kind of like a deck of cards. Pretty interesting. And we will come back to this because that's very, very, very important for our variation as a species. So again, the first phase of meiosis is called Prophase I, and the hallmark is much different. In mitosis, the hallmark was chromosomes coil, right? Well, that's still the case here. The major difference is chromosomes coil into homologous pairs, right? Synapsis occurs. That's a huge difference. Okay? All right. So let's move along and check out the next picture. Okay, so here we go. We're going to now draw metaphase I. And if you remember from mitosis, the chromosomes lined up down the middle along the metaphase plate. Super similar here. Okay, but there's something very important happening. All right, so here's a metaphase cell. Okay, and actually, you know what? Maybe we'll draw that a little bit bigger so you can see it a little better. So, okay, so here we go. We're going to draw a metaphase I cell. If you remember from mitosis, metaphase, the chromosomes lined up down the metaphase plate. Okay, they aligned along the equator of the cell. Well, in this case, in metaphase I, similar, only one huge difference here. So, we have our centrosomes that have moved to either side of the cell, and the spindle is going to be fully formed at this point for the most part. But before we do that, let's take a look at what happened here. Okay, let's say that my mom's chromosome number 1, which is now tightly coiled, is here and here. But I'm going to actually use a little blue at the bottom there. Oh, that's where it crossed over. I know that's hard to pick up on the video, so I'll try and make it a little thicker for the blue section. Okay. And then my dad's number 1 chromosome, don't forget, also crossed with my mom. So there's blue. And they exchanged those corresponding segments. Okay. So that also happened at chromosome number 2. So let's put dad on this side. Okay, so here's dad's chromosome number 2, and then, oh yeah, he has a little bit of my maternal. And I use the words mom and dad here. Don't forget, these are your chromosomes. It's just their maternal or paternal. Okay, and then here would be my mom's number 2. And we'll put a little bit of dad on there. Okay, so as you can see, crossing over actually did change up our chromosomes a little bit. We still have our kinetochores here, right? So you have your little proteins where the microtubules can grab on, right? Okay, so what's happening here is we still lined up the chromosomes down the center of the cell, but there's a huge difference. We now lined up homologous pairs down the center. Okay, so in metaphase I, a huge difference between mitosis is that homologs or homologous pairs line up along metaphase plate. Okay, now there's one other thing that's super important that happens here, and again, this might not make perfect sense yet. We're drawing a 2n equals 4 cell here. We only have 4 chromosomes in there, but in actuality, don't forget, you would have 46 chromosomes. Okay, so 23 pair lined up in a human here. Okay, now this is so cool. Okay, once we get into genetics, but there's something going on here called independent assortment. Try to wrap your head around this. What this means is that I lined these up like this. Well, mom, dad for number 1, but then number 2 was dad, mom. But don't forget, I could have also set it up mom, dad, or dad, mom, or dad, mom, dad, right? You actually, it's 2 to the haploid number, so for a human it's to the 23rd. You have about 8 million ways that you can set this up. So a 2n equals 46 is a human, right? That means that humans have roughly a 8 million, it's not quite that number, but 1 in 8 million shot of setting up this cell in a certain way, right? And that's because we have 46 chromosomes, 23 pairs. And how do you know that you didn't go with your 23 pairs like mom, dad, mom, dad, mom, dad, mom, dad, mom, dad, dad, mom, dad, mom, dad? Well, okay, right? So there's lots of different ways you can set it up. And at this point in the video, that might not make a lot of sense, but we're going to come back to it later. We've now hit our second, okay, major way that we have a lot of variation that occurs due to meiosis. The first was in crossing over in Prophase I. We shuffled up our chromosomes in ways that might have never been done before. And now in Metaphase I, we line up our chromosomes, okay, and we don't control this. This just happens that, oh, one goes on this side, one goes on this side, and that happens for all 23 pair. And there's about a 1 in 8 million shot of doing that a certain way, okay, roughly. Okay, so let's find out what the next step has in store. Okay, here we go in anaphase I. Now, this is also going to be very different from mitosis because in mitosis, we separated chromosomes at the centromere to create daughter chromosomes. We don't have individual chromosomes lined up right now. We have homologous pairs. So it's going to be a little different. And yeah, sure, I'll draw this maybe a little bit more long. Okay. And what we had in our last picture was we had mom's number 1 up here. And then we had, I believe, mom's number 2 down here. And we had crossed over a little bit of dad there and a little bit of dad there. And that means we have dad's number 1 way up here. Notice I'm drawing them apart from each other now. And dad's number 2 down here. So there's dad's number 1 and here's dad's number 2. Okay, so what actually happened here was the chromosomes, just like in anaphase of mitosis, in anaphase I of meiosis, they're moving towards the poles. Right, so here's my centrosome, and we have all microtubules coming out of there. And again, they're still connected to those kinetochores. And the chromosomes are heading this way, right? They're going towards the poles. What did we separate here? We did not separate chromosomes. We separated pairs of chromosomes. So a really important thing to remember is what happens in anaphase I, or what's the hallmark of anaphase I? Homologs separate. Okay, that's the key. And again, just like in mitosis, why did I draw my cell a little bit longer? Well, because what happens is the non-kinetic core microtubules, don't forget kinetic cores are where the spindle fibers attach. These that aren't connected to the spindle, or I'm sorry, to the centrosomes, they're going to keep growing towards each other. So non-kinetic or microtubules are eventually going to meet, and that's going to elongate the cell and start pushing it away. It looks like cytokinesis is actually beginning there. Okay, so again, anaphase 1, very, very important step. What's the difference between mitosis? Well, mitosis, we separated a chromosome, and we created 2 daughter chromosomes. We still technically in this cell, if you really think about it, we started with 46 chromosomes. We still have 46 chromosomes right now, right? We just separated them. 23 go to this side and 23 come to this side. Okay. Okay, here we are at telophase 1 now. All right, and if you remember from mitosis, telophase, there's going to be cytokinesis happening. So we're going to draw that cleavage furrow again, but a little different this time. So as you can see here, okay, our cell is pinching in 2. It's an animal cell, so we could do that pretty easily. And over here, you remember I had my mom's number 1. Okay, and these might be getting a little stringier. They might be starting to fall back into chromatin a bit here. And then we had my dad's number 2 in here as well. And he had a little bit of my mom on one of his chromosomes. And then we had my mom's number 2 over here. And she crossed over with there. And again, I'm using the words mom and dad. These are my chromosomes, right? It's just a bad habit I have. These are maternal and paternal chromosomes inside of us. So this would be inside of testes where this is going on right now. And of course we have some mom on that one. Okay, so what's happening here is we basically just separated the cell here. Nuclear envelope is going to start to reform, right? And the chromosomes are actually going to start heading back to chromatin. All right, we still have centrosomes and centriole pairs in there, sure. Okay, and what you can see here is we started with a 2n equals 4 cell, right? That's what we began with today. But technically, think about what both of these cells are going to be. We started with 4 chromosomes in the cell, but once we finish cytokinesis, this is no longer a 2n equals 4 cell. That was a diploid cell, meaning mom and dad each gave 2 chromosomes to make it 4. This is technically an n equals 20. Well, I just gave you the human number. An n equals 2 cell. N equals 2. If it was a human, this would be 23 and 23. So technically, we started with 4 chromosomes in a cell, and after telophase 1 or after meiosis 1, we're going to be left with haploid cells. Now, these haploids are no good for reproduction, and the reason for that is you're allowed to give exactly 1 copy of each gene to your offspring. If we gave these haploids to our kid, take a look at that chromosome right there. 1, 2 sides, sister chromatids, that would be giving 2 copies of every gene, right? So we can't do that. So we're going to have to go through meiosis 2 to separate the sister chromatids from each other and create daughter chromosomes. Now, we go directly, okay, these cells, once they split, are heading right to meiosis 2, right? We can't go back through, interphase again. We don't want to go through G1, S, G2. We don't want to copy chromosomes. We just spent the time, reducing the number from 46 to 23. Why would we copy it back up again? Right? So now it depends. We're doing this as a male right now. If it's a female, you can be stuck at this phase for a while. But point is, we're going to go ahead and jump right into meiosis 2. And here we go. Okay, here we go with meiosis 2. So we just split, and we have our 2 now haploid daughter cells, right? And inside of these 2 cells, if you remember, we had mom's number 1 over here that had some of my dad on it, and then my dad also had his number 2 over there, and over here it kind of looks like this, so let's fill in the rest of it. So my mom's number 1, yep, just a bit of my dad's DNA on there, and my mom's number 2, just a bit of my dad's, and now here comes my dad's number 2 that has just a bit of my mom on there, and then over here is my dad's number 1, which has a bit of my mom up there. Okay, so what happens here is, well, sure, I kind of drew them a little big here, but nuclear envelope starts to break down, right? Prophase I. So these were chromatin, and now they started to coil up. And you know what's going to happen in here is an early mitotic spindle is going to start to form. Okay, so you have your little centrioles, and they're starting to create a little spindle inside of these cells again, just like before. So prophase II is really kind of just like mitosis. The only difference is that you're starting with half the number of chromosomes, right? Okay, so let's take a look here, and if you think about it, we already started with haploids because these cells are both n equals 2, n equals 2. Okay, and if this was a human again, it would be n equals 23. So let's see how we can make true haploids that can be used for sexual reproduction. Okay, so next up, let's do Metaphase II. All right, and this is probably pretty obvious to you what's going to happen here. So we'll draw these cells here. And now what's going to happen on one side? Well, over here we have my mom's chromosome pretty fully coiled now. And then my mom's number 2. So there's my mom's number 1 and 2. Put a little bit of dad on here, right? And a little bit of dad over here. And now we're going to draw my dad's number 1 and my dad's number 2. And because they crossed over, a little bit of mom on this side, a little bit of mom down here. Okay. Now, how are these lined up this way? Well, of course, our spindle. This is metaphase after all, so you know that they're lined up right down the center of the cell. At the kinetochore, you're going to have the spindle joining, right? And that lined them right up down these metaphase plates. And of course, you have other fibers coming out all over the place as well, because anaphase is about to begin, and that's where the cell can start getting longer. So really, that one's pretty simple, right? Metaphase II, you just lined up your chromosomes. And if you think about it, there's still 1, 2 chromosomes in each cell. All right. All right, here we go. Let's try anaphase II. So just like before, maybe here I'll draw them a little bit longer. Okay, maybe that's a bit too much. But this is an important step. Think about what's about to happen. Just like this is basically mitosis with half the amount of DNA. So just like in mitosis, we separated at the centromere. Well, here's meiosis, and we're about to separate at the centromere. So technically, this chromosome right here is about to get ripped in half. So we're going to have mom's number 1 heading that way, and mom's number 1, oops, a little bit of my dad on there, right? And then we're going to have my dad's number 2 going this way, and a little bit of my mom on there. Okay. And now over here, I can see we're going to have my dad's number 1 getting pulled this way, and a little bit of my mom right there. And over here, we're going to have my dad's number 1 getting pulled that way. And here we'll have my mom's number 1 getting pulled. Just a touch of blue on there for my dad. And then over here, we have mom's number 2. Okay. Now then. Oh, geez, look what I did. Can you believe that? If you're colorblind, well, anyway, I switched to caps. Here we go. We have the spindle, of course, pulling them apart. You already know this, right? So they're getting pulled toward the centrosomes, right? So the chromosomes again are moving towards the poles, right? And you know, of course, there's other spindle fibers, and that they're growing towards each other, and that's what's elongating the cell here, right? So the non-kinetochore spindle fibers are pushing the cell apart. But think about what just happened here. We literally just had 2 chromosomes in each cell. And how many do we have now? 4, right? Technically, each one of these cells now has 4 chromosomes. Okay? Now that would be going back to normal. We started with a 4-chromosome cell. What are we doing here? Or if this is a human, this would have 46 chromosomes again. Ah, that's okay though, because these are daughter chromosomes. Okay, that's really important because these are daughter chromosomes. All right, that means that they just have one copy of every gene on them. Okay? We started with a double-stranded chromosome and we pulled them apart. And when did anaphase II begin? Well, you know that. As soon as we broke apart the sister chromatids at the centromere is when anaphase II began, and it will continue until they stop moving. Okay, so very, very important phase there. And let's see what's going on next. Okay, so here we go. Telophase II. We now have these cells, okay, that are kind of getting ready to pinch off here. All right. Now what's going on inside of these cells? Well, you already know what's happening in this first nucleus here. I have my dad's little number 2 in here. And over here I have my dad's little number 2 with just a touch of my mom's or maternal chromosome on there. And over here I have my mom's number 1. Right? And over here I have my mom's number 1, with just a tiny bit of my dad's genes on there from crossing over. And then over here I have, well, my mom's number 2 with just a tiny bit of my dad's paternal chromosome on there. And now I have my my dad's number 1 with a little bit of my mom on there. Okay, and then over here I just have my dad's number 1. And notice I'm drawing them starting to uncoil back to chromatin. And my mom's number 2. Now what's happening here is, sure, we're reforming a nucleus around them. And now if you look very carefully, how many chromosomes are in each of these cells? Oh, get out of here. Look at that. This one right here has 2. This one has 2. This one has 2. This one has 2. We went to 4 cells, each with 2 chromosomes. So that's telophase. You can see cytokinesis. Here's that ring of actin again. That's going to split it in half. Okay, so let's just show our final results here. So now, talking gametogenesis here, right? We're actually finishing this off and making these 4 genetically dissimilar daughter cells, right? And there's 4 of them. And it's really cool because if you think about it, well, let's just pick one. Let's pick the first one on there. If this guy won, what's going to be inside of this sperm? Well, my mom's chromosome number 1 stayed intact here. All right. And then my dad's number 2 stayed intact. What about this sperm if this guy won? Well, in this case, we have my mom's chromosome number 1, but that also had crossed over a tiny bit with my dad. And now we have my dad's number 2. Sure, but that crossed over a little bit with my mom. Oh, that's a very different cell right there. Okay. And now what about this cell here? Well, same thing. I have my dad's number 1 in there all folded up. But now, or uncoiled I should say, a little bit of my mom. And then we have my mom's chromosome number 2 in there, but it has a little bit of my dad on there. All right, so that's different. So these 3 are all different. And now over here, my dad's number 1 stayed intact, and my mom's number 2 stayed intact. Wow. Okay, so we actually did make 4 genetically dissimilar cells here by the end of meiosis, and all of these can be used for sexual reproduction. Okay, so that is meiosis. Let's talk a little bit more about the variation. Okay, here we go. So I just wanted to talk to you a little bit about genetic variation. Now, the main form of genetic variation comes from, some of you probably already know this, mutations. Right? We'll get into those later. That's not what we're talking about here today. But yeah, mutations, super, super, super important. okay? But we actually covered two, technically, I guess three eventually, ways that we are very varied just because we're sexually reproducing organisms. So the first way that we talked about today really was crossing over. When you crossed over in Prophase I, nobody has any control over that. Okay. Let me show you. Here's our Prophase I picture again. We have no control. We don't know where these things are going to shuffle. Okay, so crossing over is completely just a random way that when synapsis happens and these shuffle back and forth between them, if you think about where we ended up, I mean, look, we ended up with 4 genetically dissimilar gametes. The reason partly that we don't know what's in there is because we crossed over, we shuffled that deck of cards or those chromosomes, and we could technically have chromosomes with alleles on there that have never existed on planet Earth before, right? If you think about what you're actually doing is when you are going to reproduce, you're giving your DNA, yes, but what you really did was you shuffled your mom and dad's DNA in Prophase I, right? This is you, but you have 23 red chromosomes, think of them that way, and 23 blue chromosomes. And what are you going to give to your kid? Only one of each. But before you separate them out, okay, and do all of this kind of thing, you actually shuffle first. So you're technically giving your kid something that might have never existed before, okay? You have, 6 billion bases of DNA to mess around with there. So you probably shuffled in novel ways throughout your 46 chromosomes. Pretty crazy. So the first way that, besides mutations, that we talked about today that variation comes from is crossing over. And again, it happens a few times per chromosome pair. It's hard to judge where it'll be, and it could happen more than that as well. The second one is a little trickier to understand. I said we'd come back to this. Independent assortment. And there's this monk that we're going to talk about soon with genetics named Gregor Mendel, the father of genetics. And he kind of figured out independent assortment before he even knew what chromosomes were. He actually didn't know what chromosomes were at all, but he figured out that, wow, these genes kind of act on their own for the most part until you break that rule someday. So let's take a look here at independent assortment. So when we drew our Metaphase I cell, I told you that technically there are mom, dad in this case, and dad, mom for chromosome 2, right? Well, if we drew all 46 chromosomes in 23 pairs, I told you there's about 8 million different ways that you could do that. So what this independent assortment is really saying is, oh yeah, you have about a 1 in 8 million shot of making the same type of sex cell. In other words, every 8 million sex cells, they theoretically are all different from each other. That's a lot of variation, okay? And of course, that's not counting this. So if you think it's a 1 in 8 million shot, oh gosh, no, it's way, 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 way more than that, okay? Because crossing over means, yeah, you had a 1 in 8 million shot of lining up your chromosomes in the exact same way, but how do you know where they crossed over? You don't, right? So 2 sources of genetic variation so far is crossing over, Prophase I, independent assortment in Metaphase I, okay? But the third one's kind of interesting. So let's just write that on here so we don't forget. Prophase I, very important. Metaphase I, very important. Now what's happening in your anaphase I is when you separated the pairs, right? And then telophase I, the nucleus reformed. Let's say we're all done meiosis. We're left with our 4 dissimilar haploid gametes, okay? So let's go back into here again and actually say what these things are. Every one of these cells is actually an n equals 2 or n equals 23 for a human. So let's talk about that. If you really have a 1 in 8 million shot, forget crossing over for a second, but if you have about a 1 in 8 million shot of creating a specific gamete, right? Well, don't forget, this whole thing is for sexual reproduction, right? So number 3 is random fertilization. Think about this, okay? Forget crossing over for now. Independent assortment. There is a 1 in 8 million shot that that sperm is a very specific kind based on how I lined up my chromosomes in Metaphase I, right? So I have about a 1 in 8 million shot that I know exactly what's in that sperm. But random fertilization says, oh, don't forget, it takes 2 to tango. There's also an egg involved here, folks. And that egg also is a gamete, and girls had about a 1 in 8 million shot of making that exact egg. Okay, that looks like 1/8, but it's 1 in 8 million, 1 in 8 million. So technically, when random fertilization happens, sperm, egg come together, deposits the DNA, bam-o. We have a zygote. Okay, is this zygote then, well, first of all, it's diploid, right? So in this case, let's say a human, 2n equals 46. What are the odds that we got a very specific zygote? Technically, it's about 1 in 8 million times 1 in 8 million. It's about a 1 in 64 trillion shot of getting that exact zygote based on if the dad set these up in a certain way and the mom set these up in a certain way to create their gametes. So what I like to ask my kids is, does that mean that the same 2 people that made you technically had about a 1 in 64 trillion chance, if you have a brother or a sister, of having the exact same kid, the exact same genetic copy of that kid? And the answer is, of course, not even close. Because if the same 2 people that made you really had a 1 in 8 million shot here and a 1 in 8 million shot of making this egg, what are we forgetting? Crossing over, right? We have no idea when or where that's going to happen. So this 1 in 8 million and 1 in 8 million, those numbers are kind of silly, right? Because we don't know where the crossing over events happen. So technically it would be a lot more than that. Okay. So besides mutations, 2 things that happen during meiosis today are crossing over and independent assortment. And then after you've already made your gametes, sure, then you have to use them for sexual reproduction. So random fertilization, another source of genetic variation. And that's why we're probably still on planet Earth today. We're a very healthy species, well, somewhat, because of all the variation that exists. Okay? Okay, so let's just recap one more time what we learned today. We did meiosis. We created haploid gametes, right? Sex cells. We started with somatic body cells, and we turned 1 somatic body cell, 1 diploid somatic body cell, into 4 genetically dissimilar haploid gametes. Very different than meiosis, or I'm sorry, than mitosis. So meiosis makes gametes, mitosis made just regular somatic cells. So we started with prophase. We doubled up here, okay? We have 4 sister chromatids in a row, so homologous pairs come together, and that's called synapsis, and they do cross over in prophase I. Very, very important. Okay, so meiosis 1 kicks off with prophase I, very important phase. We headed into metaphase I, and this is where those homologous pairs are now fully coiled, and they line up down the center of the cell, and there's so many different ways that you can actually line these up, okay? And that's called independent assortment. We don't control where we're going to put these chromosomes. Okay, so that's another source of variation. Then we went into anaphase I, and that's where the chromosomes that were just lined up down the center, they split apart. Okay, and finally into telophase I, and here we can see that, okay, the chromosomes did split to both sides of the cell, and now the nuclear envelope is starting to form around them again. Okay, and notice that these are technically haploid now, but you couldn't give these to your kid because you'd be given 2 copies, right, of each chromosome. We have to split these now. We have to separate them at the centromere and only give 1 copy. So no cell cycle, right? We're not going back to G1 or interphase. We're going right to meiosis 2 here. And in meiosis 2, we start with prophase 2. Sure, the chromosomes coil up. The spindle starts to form. Metaphase 2, the chromosomes line up down the center of the cell. Anaphase 2, oh baby, there it is. We separated them into sister chromatids no more, daughter chromosomes, right? And now we're back to 4 chromosomes per cell, but that's okay because we're about to do telophase 2, and in telophase 2 we go ahead and we split these. And if you notice, depending on where crossing over and how we line them up in Prophase I and Metaphase I, we ended up with 4 very different gametes here. And I only drew 4 chromosomes with, one crossover event between the 2 pairs. There would be way more crossing over events in your cells, right? Because you have 23 pairs and they're crossing over like crazy. Okay, so that's how you make gametes, and I hope that was helpful.